Dear students, the topic of this session is the types of ribonucleic acids or RNA. So in cell, there are different types of RNA molecules which can be classified depending on their biological role and secondary structure. Among all of these, only one type of RNA codes for functional protein. It is called as coding RNA or mRNA, where M stands for messenger RNA. All the other types of RNA can be doesn't code for any protein, so it is grouped as a non-coding RNA. There are different types of non-coding RNAs. Few are extremely important and always available in cells. These type of RNAs are known as housekeeping RNA. And suppose, for example, tRNA or the transfer RNA, rRNA or ribosomal RNA, all are this kind of housekeeping RNA. Rest types of RNA are small nuclear RNA or snRNA, microRNA or miRNA, which actually take a very important role in the regulation of expression of genes. First of all, you can see here that although all these RNAs, the primary structure are same, but depending on their secondary structure and function, they're significantly different from each other. You can see in this slide that this is the mRNA, the messenger RNA, which actually encodes protein, whereas the ribosomal RNA or rRNA, it is a very important structural component of ribosome. Ribosome is the structure which is actually considered as a maker of proteins. Next, you can see the transfer RNA, the secondary structure of transfer RNA. This transfer RNA is actually act as adapter between mRNA and amino acids. Now, first of all, here we will focus on the three main types of RNA, that is messenger RNA or mRNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. First of all, we'll talk about the role and biological function of mRNA or messenger RNA. The name itself carries its biological function. Why it is called as messenger RNA? All the eukaryotic cells has membrane-bound nucleus. You can see here. And the DNA resides inside the nucleus. DNA carries all the informations or the blueprints of all the protein of the cell. But physically, the synthesis of protein happens inside the cytosol, inside ribosome, which resides in the cytosols. All these small structures are actually ribosomes. And here actually, these ribosomes are the maker of the protein. What happens here? Cellular machinery creates a copy of that sequence which is encodes in DNA of a protein in the form of RNA. So what is happening here? That the cellular machinery are creating a copy of the codes of a protein in the form of RNA. And this particular transcript is known as messenger RNA. And this particular biochemical process of conversion of the conversion of the DNA to RNA, mRNA, is known as transcription. This RNA transcript as mRNA is then transported from the nucleus to the cytosolic ribosome with the instruction of all the protein synthesis. Now, the ribosome reads all these instructions and synthesize a functional protein inside the cytosol. And this particular biochemical process of synthesis of functional protein from the messenger RNA is known as translation. Now, the transfer RNA next will now I'll see what is this actually. The messenger, few more informations regarding the messenger RNA. Messenger RNA carries the genetic code. Therefore, the messenger RNA carries the genetic code from DNA in a form that can be recognized to make proteins. The coding sequence of the mRNA determines the amino acid sequence in the protein produced. Once transcribed from DNA, eukaryotic mRNA briefly exists in a form called precursor mRNA or precursor mRNA, pre-mRNA, before it is fully processed into the mature mRNA. This processing step, which is called RNA splicing, removes the introns, that means the non-coding sections of the pre-mRNA. There are approximately 20,000 mRNA encoded in human genome. Next, we'll see the biological function of another type of RNA, that is the transfer RNA, or in short, tRNA. So, the tRNA actually resides in cytoplasms. Its function is to gather the proper amino acids to the ribosome during the process of translation. Ribosome use all these amino acids as a monomer to make functional protein. All these monomers to make functional proteins, which is encoded in the form of mRNA. So, ribosome needs the help of tRNA during translation. tRNA is actually picking the appropriate amino acids and delivering it to the ribosome one by one. And then the ribosome is actually stitching all these amino acids through the peptide bonds, forming the peptide bonds to form a functional protein. Next, few more informations regarding the structure of transfer RNA. tRNA is a small molecule, approximately of 18 bases, which bring amino acids to the ribosome during translation of mRNA to protein. During translation process, the transfer RNA is held responsible for choosing the correct protein or amino acids required by the body. This is also called as soluble RNA, and it forms a link between the messenger RNA and the amino acids. So, this is the secondary structure of the transfer RNA. This particular type of structure model is known as clover leaf model. Here, the transfer RNA is actually the information adapter molecule. It is direct. It is a direct interface between amino acid sequence of a protein and the information in DNA. Therefore, it decodes the information present in mRNA. There are more than 20 different tRNA molecules consisting of near about 80 nucleotides, 75 to 95 nucleotides may be the range. The clover-leaf model of tRNA is secondary structure consists of 4 stems and 4 loops. You can see here 4 stems, 1, 2, 3, and 4, and 4 loops, 1, 2, 3, and 4. The 4 stems are actually double helix, stabilized by hydrogen bonding between the bases, and 3 of the 4 stems have loops. This kind of loops they have, containing 7 or 8 bases, at the ends, and the remaining unlooped stem contains free 3' hydroxyl and 5' ends of the chain. The unlooped stem is called as the acceptor arm. This is actually the acceptor arm. So, actually, this is representations of the secondary structure of the tRNA. Now we will see about the what is ribosomal RNA or rRNA. So ribosomal RNA is a main component of ribosome, where ribosome can be considered as protein synthesis machine of self. Here, ribosomal RNA bind with stem protein and form ribosome. So, the ribosomal RNA is a component of ribosome and located within the cytoplasm of a cell where ribosomes are found. In all living cells, the ribosomal RNA play a fundamental role in the synthesis and translation of mRNA into functional proteins. So the ribosomal RNA is mainly composed of cellular RNA and at the most predominant RNA within the cells of all living beings. Next, quickly we will see the different kinds of regulatory RNAs. First of all, the small nuclear RNA. What is small nuclear RNA? In abbreviation, it is known as snRNA. Small nuclear RNA are always associated with a group of specific protein to form the complexes referred to as small ribonucleoproteins. Ribonucleo because the presence of RNA and protein, it is forming complex with protein. So totally it is a ribonucleoprotein in the nucleus. Their primary function is to process the precursor mRNA. Next word is another kind of regulatory RNA present inside the cell, that is microRNA. In abbreviation, it is also called as miRNA. miRNA are small non-coding RNAs of near about 20 to 22 nucleotides and the most widely studied classes of non-coding RNAs. These RNAs mediate post-transcriptional gene silencing through the RNA interference. The mechanism is very important. It is called as the post-transcriptional gene silencing through the RNA interference, where an affected complex of miRNA and enzyme can target complementary mRNA by blocking the mRNA from being translated or accelerating its degradation. That means, in very simple way, this short, small stretches of nucleotides actually interact or binds with mRNA and blocks its translation to the functional protein. Otherwise, it can also accelerate its degradation. In human, miRNA are estimated to regulate the translation of more than 60% of protein coding genes. So this session is up to this. These are the reference. And finally, thanks to all of you.